Welcome to today's lecture on mitochondrial dysfunction. We will explore how mitochondrial diseases affect cellular function, focusing on electron transfer, ATP synthesis, reactive oxygen species, and cellular adaptation to stressors. Join us to gain a comprehensive understanding of these molecular mechanisms and their implications. Complex I, or NAD-ubiquinone oxidoreductase, is vital in oxidative phosphorylation and ATP synthesis. Mutations in complex I lead to mitochondrial diseases, like Leigh syndrome and Leber hereditary optic neuropathy. It consists of subunits and cofactors that, if disrupted, impair electron transfer. This leads to reduced ATP production and increased reactive oxygen species. In oxidative phosphorylation, iron-sulfur clusters play a key role in mediating electron transfer. Dysfunction in iron-sulfur cluster assembly can lead to mitochondrial dysfunction in various diseases. Maintaining iron-sulfur cluster balance is important for mitochondrial function and preventing oxidative stress. Proton leak, or uncoupling, allows dissipating energy as heat instead of ATP. Uncoupling proteins, UCP1, UCP2, and UCP3, regulate energy expenditure and thermogenesis. Dysregulation is linked to obesity and altered metabolism. Targeting these mechanisms may lead to therapeutic strategies for metabolic disorders. In this section, we will discuss how the ATP synthase enzyme complex synthesizes ATP in oxidative phosphorylation. Factors such as ADP and inorganic phosphate availability, proton gradient, and regulatory proteins influence its activity. Dysregulation can lead to energy depletion and is implicated in diseases. Understanding this regulation could help develop interventions for mitochondrial diseases. Mitochondrial DNA, mTNA, encodes important components of the electron transport chain complexes. Mutations in mTNA can disrupt their assembly and function, impairing oxidative phosphorylation. These mutations often show maternal inheritance and can cause various symptoms affecting multiple organs. The replication, repair, and interaction of mTNA with nuclear DNA are key factors in the development of mitochondrial diseases. In response to problems with mitochondria, retrograde signaling pathways send signals from mitochondria to the nucleus. This alters gene expression, allowing cells to adapt to mitochondrial dysfunction. Failure in retrograde signaling can worsen mitochondrial dysfunction and contribute to mitochondrial diseases. Reactive oxygen species, ROS, are produced during mitochondrial respiration and have important signaling functions, but excessive ROS can be harmful. Antioxidant systems in mitochondria, like superoxide dismutase and catalase, counterbalance ROS levels to prevent oxidative stress linked to diseases like neurodegeneration and cancer. In the video, we will discuss how cells respond to low oxygen levels during hypoxia by reprogramming their metabolism. Mitochondria play a vital role by adjusting metabolic fluxes. The HIF pathway is a key player in this adaptation, promoting glycolysis and inhibiting mitochondrial respiration. These changes are crucial for cell survival, but if not regulated properly, can lead to hypoxia-related diseases. The mitochondria and endoplasmic reticulum, ER, work together through specialized contact sites called MOMs. They exchange lipids, calcium ions, and signaling molecules, which are vital for cellular processes. Problems with MOMs and mitochondrial ER communication are associated with diseases like neurodegenerative diseases, metabolic disorders, and cancer. Mitochondrial quality control is essential for cellular well-being. Dysfunctional mitochondria can be eliminated through mitophagy, a process involving degradation via lysosomes. Dysregulation of mitophagy and impaired mitochondrial quality control contribute to diseases like Parkinson's and aging-associated disorders. Studying mitophagy mechanisms can aid in developing treatments to improve mitochondrial quality. Cancer cells have a unique metabolism called the Warburg effect, where they rely more on glycolysis than oxidative phosphorylation. However, mitochondrial function still plays a crucial role in cancer progression, metastasis, and resistance to therapy. Understanding how oncogene signaling, metabolic reprogramming, and mitochondrial function interact is an exciting field in cancer research. Oxidative phosphorylation is a process in which cells generate ATP, the energy currency of the cell, through the transfer of electrons along the electron transport chain. This process occurs in the mitochondria, the powerhouse of the cell. During oxidative phosphorylation, electrons derived from fuel molecules are transported through a series of protein complexes in the inner mitochondrial membrane. As electrons move through the electron transport chain, energy is released and used to pump protons across the inner membrane, creating an electrochemical gradient. The resulting proton gradient drives the ATP synthase enzyme to produce ATP by combining ADP and inorganic phosphate. This process, known as ATP synthesis, is essential for cellular function. Redox signaling refers to the communication between cells mediated by redox reactions. It plays a vital role in cellular adaptation to environmental stressors such as toxins, radiation, and oxidative stress. Redox signaling involves the balance between oxidants, reactive oxygen species, and reductants, antioxidants, in the cell. Cells have intricate antioxidant defense systems that protect against oxidative damage caused by an imbalance in redox signaling. However, excessive oxidative stress can overwhelm these defense mechanisms and lead to cellular dysfunction and disease. Understanding the mechanisms of redox signaling and cellular adaptation to environmental stressors is essential for developing strategies to enhance cellular adaptive responses and mitigate the effects of these stressors. Understanding the intricate mechanisms of oxidative phosphorylation and ATP synthesis, including complex I dysfunction, proton leak, and mitochondrial ER interplay, is crucial for addressing human health challenges and creating targeted therapies.